We'll start now. Good morning, one and all. I am Dr. Sandeep Rao, Innovation Manager at Innovation and Incubation Center for Entrepreneurship, also known as IISER Bhopal. Thank you all for sharing your time, and I invite you for the Entrepreneurship Talk by Dr. Vishal Rai. IISER Bhopal has recently started the Virtual Entrepreneurship Talk series, to encourage and inculcate the entrepreneurial spirit among the students, researchers, and faculty members. And today's talk is the third chapter of the same series. Today's talk is also special for ICE for two reasons. One, Dr. Vishal Rai is the first faculty entrepreneur of IISER Bhopal, and he is also the first startup incubated at ICE Incubated. So while he'll be talking about the journey of his company, Plabeltech Private Limited, it will also partially touch the journey of ICE Incubator. And there is another reason which makes this talk special. ICE organizes the institute level entrepreneurship competition called as Curveball every year to select potential student ideas as student pre-startup at ICE. Last week, we had a nail-biting pre-qualified round of this competition, and 5 out of the 9 teams made their ways for the finals. These finalist teams are undergoing mentorship at ICE for their preparation for the final round. And what else can be a better mentorship than the faculty entrepreneur sharing his own experiences with this team? So we hope that today's session will also help our finalists and will make the curveball competition better. With this, I would request all the participants to be active throughout the session. The objective of today's talk is to hear from the students, researchers, and faculty who also aspire to be an entrepreneur. And whatever the questions or concerns you have, you can share those questions in the chat box. And at the end of the talk, we'll try to accommodate as many questions as possible. I'll also request all the Zoom participants to please keep yourself muted and help us in a smooth conduct of today's event. With this, without any further ado, I request Dr. Amjad Hussain, CEO of IISER Bhopal, to formally invite and introduce our today's guest speaker. Over to you, sir.
Thank you, Sandeep. Can you hear me? Am I audible?
Yes, sir. You're audible.
Thanks, Sandeep. And thank you all who have joined for today's talk. It's really a privilege having Dr. Vishal Rai as today's speaker for the third Entrepreneurship Talk, which is a part of the series started by ICE, which is a virtual entrepreneurship talk to encourage entrepreneurship and innovation at the IISER Bhopal campus. Dr. Vishal Rai, everyone knows that he's an accomplished scientist, and he is an associate professor and Swarnajayanti Fellow at the Department of Chemistry at IISER Bhopal. He has started his scientific career with a PhD in chemistry from IIT Bombay, and followed by a postdoctoral position at University of Toronto in Canada. He also got selected for a position at MIT ACS Elevate Fellow, which was also in Canada. And then subsequently, he has joined IISER Bhopal as an assistant professor. At IISER Bhopal, his research group is leading the development of chemical technologies in the precision engineering of the protein. His group is also involved in the synthesis of homogeneous antibody drug conjugates, protein immobilization, and analytical tools for peptides and proteins. And his research team wants to contribute towards the society by enabling directed therapeutics and making precision therapeutics possible in the future. And I'm sure those who know about antibody drug conjugates, this is one of the novel technology which can be used in synthesis of antibody drug conjugate for precision targeting of cell and cancer cell, which is pretty much in potentially used in other countries and now getting popular in developing countries as well. Vishal is also the founder and director of Plabeltech Private Limited, as Sandeep very rightly mentioned. His startup was the first startup incubated at Innovation and Incubation Center for Entrepreneurship at IISER Bhopal. His company is already making a good progress and is empowered by state-of-the-art protein engineering technologies such as LDM-TM, GlyTag-TM, and mass spec TM. Dr. Vishal is also a recent recipient of Swarnajayanti Fellowship, also has been a recipient of Ramanujan Fellowship and DAE Young Scientist Award. Very recently, he joined the ACS Chemical Biology team as an early career board member. Also, he is invited to become a fellow of the Royal Society of Chemistry, FRSC, United Kingdom. Dr. Vishal is an accomplished scientist, a brilliant teacher and entrepreneur, and it is really a pleasure having Dr. Vishal today at ICE to deliver the third entrepreneurship talk. I'm very pleased to invite him for the talk. Welcome, Vishal. We look forward to your talk. Thank you.
Thank you, Dr. Amjad, for very kind and elaborate words of introduction. Let me just share my slides quickly. Okay, can you see my slides right now?
Yes.
Okay, thanks a lot. So let me just minimize this part. Okay, so welcome everyone. At the onset, let me thank Dr. Amjad and Dr. Sandeep for giving me this opportunity. I hope all of you are holding strong and staying safe while tackling the ongoing pandemic. In your academic life, you would have already realized that the decision-making process is something that you have to exercise every day. And research life is not very different in that perspective. So in today's talk, I'll use the example of my research journey to highlight how the entrepreneurial filters can be highly valuable when it comes to the decision-making process for the selection of research problems. Alongside, I will also highlight what Plabeltech is doing at the Incubation Center of IISER Bhopal. Now, when I had to start my independent research journey, I was constantly asking myself that what would be my big research question. And there are two general ways to go about such type of decision-making process. You can develop an interesting method or technology through an exploratory route and then look for its potential translation depending on the kind of market that you can address based on what you have been able to find. Alternatively, you can initiate by looking at the technological demand first, then you can do a retro-analysis and try to develop a solution for that. Now, both the options have their own merits and demerits. So the first option is what I practiced during my PhD and postdoctoral research. And I had a mixed experience in this approach where we had one failure and one success in the translation of our scientific methods or technologies to the market. Now, I'm more convinced by the second option, and that's why for my independent research career, I was aggressively looking for the technological demand-driven research question by the time of around 2010. And this was also the point where a lot of other research activities were happening in my life that kind of dictated on how I ended up being in this particular research problem that we are looking at this time. So let me give you a little bit of flashback to the story and tell you about a few important characters who were a very important part of my life at that time and probably also responsible for what led to the selection of the question that I am working at currently. So 2010, was the time when I was enjoying my postdoctoral research with Professor Andre Yudin, who can be seen in this photograph over here. He's an amazing scientist and a really great human being. And we're working together. And Ryan, who is currently a faculty at York University in Canada, so Ryan and I were trying to find a cyclic peptide inhibitor for a very specific protein-protein interaction in a collaborative project with Professor Tony Posson. Now, the scientific details are not important for today's talk. However, it was really intriguing that with all the structural data from NMR, support from computational team, and some of the best in business on board, we were still not able to get to the exact solution that we were looking for. And at this time, it was clear that in the multiple steps happening inside the cell, something is not going as per what we expected or designed. So it left me wondering that, okay, why is it so that we have to work with this black box type of system? So why can't we place a probe that can report back to us about what is going right and what is going wrong? And this is when I realized that there are no chemical methods that can deliver precision and modular engineering of native proteins. In fact, when I discussed it with some of the team members, I realized that the general perception is that such methods are not even possible. So this is why I decided to see if we can develop chemical methods that can empower us to deliver precise single-site modification of native or unengineered proteins. Now, at this point, an obvious question should come to your mind that, okay, is this an exploratory research question, or is it a technological demand-driven research question? Now, since this is not an interactive session, I'll have to answer this question myself. So it is an exploratory research question that is posed by a well-defined technological demand. In other words, it's a mix of both. So let's see what technological demands can be met if we can answer this question successfully. So the basic knowledge of reactivity and diverse aspects of selectivity with proteins provides a great opportunity to contribute to the market of biophysical and biochemical tools. Now, if you are trying to investigate diverse types of post-translational modifications, or you want to look into the interactome of a biomolecule, or you want to image a particular cellular process, or want to localize a biomolecule within the cell, you would need specific tools. Now, the fact that this segment is doing pretty well, even with the non-selective kits, tells a lot about the potential opportunities that lies ahead for the selective technologies. Now, let's look into completely different portfolio. If you know about the market of vaccines, more than 25% of the share is held by conjugate vaccines. So typically, they would involve a carrier protein that is covalently connected to polysaccharides that can serve as antigens. So you should probably note at this point that it's not really straightforward to control the chemical purity of the polysaccharides. And that is something that's really an active area of research right now. And there are some amazing contributions coming from Europe in this perspective. But besides that, it is also super challenging to control the precision or selectivity of the bioconjugation. And that again contributes to further heterogeneity. So typically, one is dealing with a very heterogeneous mixture with a lot of issues that are associated to the batch-to-batch reproducibility, even in the conjugate vaccines that are available in the market and are being administered right now. But apart from this, what really excites our research team is the potential to contribute for directed cancer chemotherapeutics. This is something that we can achieve by preparing homogeneous antibody drug conjugates or ADCs. Now, this provides a state-of-the-art technology, and as the name suggests, it involves a monoclonal antibody that is covalently connected to a toxin through a linker. Now, when these ADCs are given to the patient, the job of the antibody is to target the antigen that is overexpressed in the tumor cells, but not in the normal cells. And that is the basis of targeted chemotherapeutics. Now, in the next step, this overall complex of antibody drug conjugate and antigen, it gets gradually internalized, and then eventually it will be taken over by the lysosome, where the lysosomal degradation is going to release the toxins that eventually would lead to the apoptosis or death of the tumor cells. Now, there are a few drugs that have received the FDA approval and are available in the market. However, these drugs have one thing in common, that they are as heterogeneous as what you were seeing in the case of conjugate vaccines. So there will be 0 to n number of toxins that will be attached to the monoclonal antibody. Now, is this a bad thing? Is this heterogeneity a bad thing? So quite systematic studies have been done in this regard, and now it is pretty well established that the heterogeneity is detrimental not only to the pharmacokinetics, it also brings in batch-to-batch reproducibility issues, and have been one of the key reasons why there were failure of multiple efforts in the preclinical stages. But now you would also realize that not much can be done to solve this problem due to lack of enabling technologies. So I'll talk about some of these aspects in the later part, but as we are working with such protein-based therapeutics, we also understand that they will not be accessible to a huge population across the globe simply due to the cost factors. And that is why, in the long run, we want to see if we can engineer a single site of a single protein inside the human body. Because if we are successful, it could provide a disruptive platform for precision therapeutics, because then small molecules can provide the specificity that has so far been possible only with the biologics, and that would be amazing. But before we can work with the complex systems such as humans or tissues or even cells, we would need the knowledge and methods that can enable precision engineering of isolated proteins. And once we have them, we can go back and translate them sequentially from one complex system to the other. Okay. At this stage, you should have probably one simple question in your mind, that do we not already have drugs that are highly specific in nature? So let's take a look at one such representative example. Kinase inhibitors are very important for several critical signaling pathways in our body. So it's really not a surprise that there are multiple companies that have been investigating it over the past several decades, and it has led to multiple FDA-approved drugs as well. In fact, in the last 1 decade, there have been more than 38 FDA-approved drugs that follow the lines of kinase inhibitors. But along with that, a lot of research groups have also started questioning the specificity of these kinase inhibitors. So the chemoproteomics allows you to now, for the first time, look into the details of what the potential targets are. And what has been very well determined now, that even the most selective protein kinase inhibitors goes for at least 30 to 100 identified targets, apart from many more unidentified ones. And this is exactly the reason why these drugs have multiple off-target activities and lead to side effects in the patients. Now, a part of our team is trying to see if we can solve such challenges in the long run. Now, there are two important points to note at this stage. The technological demand is just one part of the entrepreneurial filters. And it is critical to think about the business sustainability and the growth model when you are going for setting up a company. And this is where the translation time and cost factor becomes really valuable in the decision-making process. Now, in this perspective, the biophysical tools and biochemical tools that I discuss, they constitute a low-risk segment where the product development would take less time. Now, even though the profits will be moderate, it can help you in the sustainability of the company without really diluting the shares. Okay, and then for us, the next step comes in the form of biologics, which has a few risk factors involved for sure, and will definitely take far more time than the previous tools that I mentioned. But then once it is successful, it can boost the magnitude of company upon its translation. Besides, it can provide the resources that we need for the development of precision therapeutics in the later stage that would require a far more investment in terms of time and funds. But then it can really take the company to an altogether different level. So these are a few filters that went into the background for selection of our translational research question. However, the first step in this direction was basically related to exploratory basic science question. And in this perspective, we started by looking at protein as a multifunctional organic compound, which is full of nucleophilic residues. So for all the non-chemists in the audience, nucleophilic residues will be able to donate a pair of electrons, and then you put an electrophile that can accept a pair of electrons. So these two will react and form a bond. And that is what we utilize for the bioconjugation. So for covalent bond formation, as we discussed, we will typically add an electrophile. So all these nucleophiles are going to compete for that electrophile. And then the electrophile will have to figure out how to differentiate one type of residue from the rest. And once we find such an electrophile that is chemoselective and can differentiate one functional group from the others, and let's say it is selective for a primary amine, then there will be multiple copies of primary amines present in the protein. So the next task for the electrophile would be to see that how is it going to differentiate one primary amine from the rest. And that is what we term as site selectivity. So for precise single site modification of an isolated native protein, we would need an electrophile to exhibit chemoselectivity and site selectivity. Now let's say this also happens to be the most reactive primary amine. Okay, and we want to modify this one or this one. Okay, is it really even possible to go beyond the reactivity order that is defined by the protein, right? And this is where we really had to challenge the existing thought process in the scientific community that was practicing bioconjugation. And this is something that I would also refer to as modularity in the latter part of my talk. So with this background, we asked a very simple question that let's see if chemical methods are capable of identifying the reactivity hotspots that are present in the protein. And after our initial investigations, we learned that N-terminus alpha amine of a protein in multiple cases could serve as a preferential reaction center and can be labeled with high precision with an appropriately designed reagent. So with this in hand, we now wanted to see if we can differentiate the reactivity of one residue from its multiple copies. And we were delighted to learn that not only this is possible, but we can also regulate chemoselectivity and site selectivity with absolute precision for modification of some highly challenging residues. Besides, we can apply these methods to a structurally diverse set of proteins, install different probes of interest, and control the type of reaction or type of bonds involved in the bioconjugation. So in a simplistic language, we had a very comprehensive toolbox that allowed us to engineer a bond in the way we wanted. And it really provided us a flexibility that was unprecedented. But it is also important to note that all these methods were still limited to targeting the reactivity hotspots in a protein. And we became even more ambitious and wanted to see if we can target a single site of a single protein in a mixture of, let's say, 5 to 20,000 proteins. Solution where, let's say, I have 5 to 20,000 proteins, can I still target one single protein at a single site? And this is why we started working on a residue-specific method. And Purushottam, Unni, and some of our other team members did a remarkable job while developing the GlyTag technology that can empower us to do just that. Now, along with this, we really wanted to see if we can go beyond the reactivity order defined by the proteins and develop modular technologies. And the potential opportunities that can be accessed with such methods was a great driving force for us. Now, with outstanding efforts from Srinivas, Datta, and other team members, we were able to come up with LDM platform. And we're really proud of this technology because it works well with a structurally diverse set of proteins under physiological conditions. It empowers the installation of diverse biophysical and imaging probes and delivers analytically pure single-site tagged protein. And at this point, we were already way ahead of any other technology in the market. And we were all set to see how LDM platform would work for the synthesis of antibody drug conjugates. Now, let's have a quick recap of the challenges that needs to be addressed with the help of a short video. So, as we discussed, the ADCs are composed of three key components. The toxin is the first one, and in this case, it's monomethyl auristatin E. And then the second component will be the monoclonal antibody that you see over here. And the third component is the linker that connects the two of them. So now this would also lead to the first question, that how can we control the homogeneity of bioconjugation? Now, once this problem is solved and the ADCs are injected to the bloodstream, it also becomes important to see that what would be the bond strength that connects the toxin to the antibody, because this is a very critical parameter, as the toxin should not be released before the ADC reaches the tumor tissue. If that happens, then you can anticipate the off-target effects because the selectivity of delivery is gone. And that is why the control over chemistry and bond type becomes really, really important. Now, eventually, when these problems are resolved, the ADC is going to reach to the tumor cells, right? But they might or might not kill the tumor cells. So what decides that? This is where the control over conjugation site becomes very critical to ensure that the ADC antigen complex does get internalized, right? And once this problem has been solved to a good level, eventually the overall ADC antigen complex would reach the lysosome, as you note here in this video. And then the lysosomal degradation is going to release the toxins, which is going to be spread around, and it will eventually kill the tumor cells and would migrate to the other cells of the tumor, which will, go towards apoptosis, as you see here in this case. Now, this is exactly what our LDM technology offers in terms of promise. It provides a very powerful platform with unprecedented control to meet the demands of consistent homogeneity and drug to antibody ratio, or DAR. Besides, it offers flexibility with the chemistry and the residues of monoclonal antibody that would be involved. Also, we can regulate the bond strength, linker chemistry, and the site of conjugation. So now we have uh, ADCs that outperforms one of the marketed drugs from Roche during the initial investigations. And importantly, our ADCs are highly specific towards the antigen positive cells, which represent the tumor cells, and does not show any negative impact on the antigen negative cells, which represents the normal cells. So it meets all the initial standards that are required for taking it as a candidate for directed cancer chemotherapeutics. Now, at this point, let me tell you another short story in this perspective. So the pharma sector noticed an enormous boom in efforts towards the antibody drug conjugates for this purpose after the initial concepts were established some time back. Then why do we not have the proportional number of ADCs in the market? Unfortunately, a significant percentage of efforts failed at the preclinical stages, and the huge roadblock emerged from the structural complexity of the ADC that you see over here. Okay, now you can realize that it's not straightforward to analyze the site of drug conjugation, even with the most advanced mass spectrometers. Besides, you have to make sure that the overall composition is not affected, the glycan composition does not get affected, and that all delivers batch to batch consistency. So when you're developing these methods for the synthesis, you have to make sure that everything happens under very, very mild condition. So while LDM technology takes care of these questions and the heterogeneity, our mass spectral sensitivity boosters solve the problem of analysis to a great extent. So it enabled us to identify the site of drug conjugation with unprecedented accuracy, and we are really happy with the results that we were able to achieve with that, and we are looking forward to utilize it further. So now all the state-of-the-art technologies that I discussed, they form the foundational basis of Plabeltech. We have one product on GlyTech platform that is for the metal-free protein purification, specifically for the therapeutic proteins. We have 3 mass spectra series products for enhancing the protein sequencing and analysis of ADCs. Also, there are 3 more products in the pipeline from LDM platform. Now, along with the launch of the products that I just mentioned, we also initiated our service portfolio in this February. And within 2 weeks of launch, we completed sales of 2 products. We provided service to 1 business customer. And but yeah, at the same time, we will again have to relaunch things once the COVID-19 normalizes. So we are keeping things on hold, minimizing the investment in the company at present. Besides, we are aggressively working on our ADC portfolio and looking forward to our 2 closest competitors in this domain, that would be Cadsilla and Inhertu, that are already there in market, and they are doing pretty well. And here, both these compounds that are used, the trastuzumab emtansine conjugate or trastuzumab deruxtecan conjugate, both are heterogeneous. So there are obvious problems associated to it. And this is where we think that we would have a shorter travel path to the market and a well-prepared market for taking our products into it. So for this, we are already in final stages of discussion for partnership with the company to take our ADCs to the IND stage before we can sell it out to another pharma company for taking it all the way to the market. And with this, I guess it's time to thank all my team members. Nothing would have been possible without their immense motivation and hard work. I'm really privileged to have friends and team members who are strength pillars of our multidisciplinary research program. And I thank them all, and also the funding agencies for their constant support for all these years. None of this would have been possible without their generous support to our research group in the Department of Chemistry at IISER Bhopal. I must also thank BIRAC for the Biotechnology Ignition Grant that actually helped us setting up the company. We are thankful to the Incubation Center of IISER Bhopal, and we take pride in the fact that we kind of were born together and started our first few steps together. We are thankful to the C Camp Bangalore for their mentorship to the BIRAC program. And thank you all for your kind patience. I'll be glad to take your questions.
Thank you, Dr. Vishal, for such an informative and exciting talk. And I believe that this talk might have surely encouraged our participants. So we'll start with the question and answer session. I have a few questions in a Google Drive. Let us start with the first question, and I think it is very relevant for our today's audience. When do you think is the best time to explore science entrepreneurship? Is it immediately after the post-graduation, after PhD, like slightly understanding the stream and the technical part of it, or after postdoc and securing the future?
Well, there's no perfect answer to this question. I guess science entrepreneurship is more of an attitude that you should always have with you. It kind of helps you, as I was, you would probably have noted in my talk. That's a thought process that you can use as a filter for selection of the problems you want to work on. Typically, you would have 40 years, 45 years of research career at the most. So, and you will have a lot of questions, so you can't answer all of them. You will have to make those selections. So you can use them as filters to make selection depending on the research problem you are working on and the type of questions you are addressing. You would realize that you can yourself note that, okay, when is the right time to enter into the translational part through either a startup or through transfer of technology or a different means. So keep that attitude with you, and then move systematically and wait for the time. And this time could be, the right time could be different for different people depending on the project you're working on.
Yeah. Dr. Amjad has two questions.
Can I just put one disclaimer though? So, while I'm answering all these things, please don't take my words for the absolute knowledge in the field. There are multiple ways, multiple correct answers in the field. What I would be telling is just one possible route through my experience, but feel free to do experiments and have your own route. So you should always do what makes more sense to you. Just listen to what others are also saying, but do what you feel is right. Yeah, go ahead, Sandeep.
Yeah. Dr. Amjad, you can ask your question.
Hi, Vishal. It's a wonderful talk. I really enjoyed it. So I have two questions. The first is related to this ADC conjugate. As you know, there is an evident problem of having these ghost antibodies. So for example, if there is an antibody, but then the toxin is not conjugated with that due to either a weak linker or the failure of linkages. So does your technology also address to make sure that there are no empty antibodies, because that won't make any use of delivery?
Yeah. Thanks for raising this question. I kind of skipped on this simply to keep the technical part minimized. But this is really a very good question. So when you're making these antibody drug conjugates, whatever you will find in the market, they can maintain homogeneity only up to 10% conversion or so. So you would anticipate that at least 80% or above would be the unlabeled antibody. Now, the unlabeled antibodies, even though they are specific, they are not really toxic. So at those concentrations, they won't be really doing much except, blocking some of the receptor sites. So in our technology, though, the advantage is that we have a module developed where we can capture the antibody, and then the toxin installation is going to be happening in the late stage, releasing the antibody drug conjugate, which practically means that our ADCs would not have unlabeled monoclonal antibodies in that.
Okay.
Yeah, so it does take care of the challenge.
Excellent. And my second question is, in regards to possibilities of getting exits, because it's a life science, or I would say a biotech, heavily sort of technology driven company.
Right.
And generally, people are a little bit fearful when they would like to launch themselves as a faculty into entrepreneurship in such companies, starting up such companies, because there is a huge gestation period. So if you could shed some light on what are the possibilities of taking exits, either through translating technologies to bigger companies, or mergers or acquisitions. What are the ways where you see that Plabeltech can make revenue, or such companies can make revenue, even without touching the endpoints of becoming independent?
Yeah, no, this is a very good question again. Exit options for the company or for a specific technology, both are really critical points to evaluate for any startup. For Plabeltech, I'll tell you what we are doing. We have some biotech tools, or which I mentioned, biophysical or biochemical tools. These are the ones which we have already brought to the market, but we are not really going to take it into the sales and marketing division. So we are trying to find out or sort out the right partners who can do the sales and marketing for us, and we can simply focus on the other portfolio where we are developing new products. So that's what we want to look into at this stage. 10 years down the line, that might change, but at this stage, that is what we are going to do with the biotech tools that we are coming up with. Similarly, when it comes to anything that is related to, so the advantage is that with these tools, you have lesser time required for taking it to the market, and same thing is in terms of fund investment. But when it comes to anything that is related to therapeutics, the time would be really far more, and so will be the cost. And for a startup, it is not, at least for us, it's not a great idea to think that we can take ADCs all the way to the market. We would not have that type of deep pocket for some time. So the plan is very simple. We have one company interested that wants to do all the pharmacokinetics part, while we are doing the other chemical, analysis that is required for finalizing all the documents for the pre-IND and IND application. So investigational new drug is the stage, or IND is the stage where we are planning to out-license it to a big pharma company that would have pockets deep enough to take it all the way to the market and support the clinical phase trials and so on. So that's the exit position for us for ADC platforms. So depending on what we are working on, we have to think about it. Some of the entrepreneurs who they also prefer setting up a different company per technology. So they will one new technology would constitute one company, and they do it because they are very clear in their mind that once the technology is kind of have shown its promise, they will find the buyers and investors who will take over that company. So their exit option would be for that particular company at that stage. What we are doing is that we are not setting up multiple companies with every technology. We will be exiting for a technology rather than exiting for a company. So I hope I answered your question to some level
Yeah.
of satisfaction.
Thanks, Vishal. Thank you for answering.
Yeah, there is another question by Sweta Satpathy. What are some things that are crucial to be taken care of while securing the funding for science entrepreneur?
Sorry, what are?
What are the things which should be specially taken care while we want to secure the funding for science entrepreneurship? So how science entrepreneurship funding is different than the normal scientific fundings?
Oh, okay. Well, so normal science funding comes from funding agencies like SERB and DBT and maybe DAE to some extent. But if you look into their template versus you look into the template that comes for BIRAC Big Grant or SIBRI, these are the things that come for the entrepreneurship. They are very different. The points that would need emphasis in each of them, they are not the same. So a good way to understand that would be to just take those templates and the questions that have been asked where they want you to elaborate on certain aspects of your proposal. That itself will be a very good directing point to tell you that, okay, what is more important for the entrepreneurship and what is probably more important for a discovery based elaborative and exploratory research that can still fall under the purview of academic research projects.
Okay. There is another question by Divesh Shah, and he asked, what encouraged you to transcend the academic boundary and translate your tome of research to applications?
So, okay, this is something that I have noticed to impact the economies of either North American countries or European countries. So science has a tremendous potential. And if any country's economy has to grow, the new technological growth from the end of science has to be there. Okay. And if you think that, okay, for example, when you buy any kit, I do not know what your background is, but you can think it in term, depending on your background, you can think of a biotech kit or you can think of a car or bike, right? When you compare the technologies, there are a lot of small things that goes into every product that is out there, right? And if there are not enough people doing all these new discoveries, the cost of the product will go up depending on how many ingenious technologies have been used in the development of that product and how many imported technologies have been used, right? So at the larger scale, it affects the overall economy of the country. And there's no second thing related to this. So for us, specifically the type of research questions that we want to address, we as an academic group, I must say that in my heart I'm still an academician. It's just that I do not see the translational part or entrepreneurship as anything different from what an academician should also think about. So now it's the simple thing is that if I want to address very, very challenging questions, it would also mean that I would be competing against the best in the world. And it would also mean that the funding scenario is not going to be the same. The best funding that I will have in India from a funding agency like SERB or DST, DBT, whatever, is going to be at least 5 to 10 times lower than what my colleagues would be getting in other parts of the world, right? So how do I compete? How do I, get the things done that I want to? Well, so I would need more funds. How would that come, right? I would need alternative sources of the fund. So that is where entrepreneurship comes to me. Means I think that it could be a very important lateral financial support for the research problems that we want to solve. Besides, there is one more thing that happens automatically. When you start thinking about translation of your research problems or the questions that you are addressing, it makes you read about the connection between the lab research to the market. And there are a lot of such things that we would normally not get the time to read about if we are not thinking about the translation of those technologies. Even, for example, when I was talking about antibody drug conjugates as an academician, I would not really think about all those little details that goes into the IND application, where you have to characterize all these little things, the batch to batch variation and other aspects related to the product that you are making that you will never test as an academician, right? But then you start paying attention to those details when you are thinking from the perspective of an entrepreneur. So overall, I think it just makes you a better researcher. It gives you a much wider or broader perspective of what it takes to translate your technologies, and that's more or less it. I hope I answered your question.
Yes, sir. There is another question that this investment series A, B, and C, are entirely different compared to the grant in aid or seed funds, which the entrepreneurs initially receive. So, what is your experience? What different sort of preparation or achievement is required to go for the investment series?
So this is a question that I would prefer to redirect to Dr. Amjad or Sandeep, you, because so we have been trying to delay investment,
Yeah.
and I'll just tell that means what we want to do is that we want to take the company value up before we go to the investors, so that whatever dilution we have to do, we can demand for more investment per share diluted. Okay, so that is why we first of all want to make sure that our value is good before we start disinvesting. And this is why you would notice that we thought that, okay, if we have certain products that we can translate quickly into the market, they will start generating revenue that would be good enough for our sustainability. So we can delay the dilution or, taking the money from the investors. And in that case, we will have a better prospect, or we can get a better deal in terms of dilution from the investors. And then I'll pass this question to Dr. Amjad, that how does the investment series work?
Yeah, I'm happy to take it, Vishal. First of all, when we talk about raising funds through Series A after seed, let me tell you, there are a lot of successful companies. They do not even would like to raise Series A funds after seed funding, because the idea is that one investor puts the money in, then they expect some sort of shareholding from the company. And most of the time, when you are raising funding, it is on their terms and conditions. So, and then a lot of founders, they keep diluting their shares through Series A and Series B, and then eventually they end up having nothing. But if you do it strategically, it also adds to the development of a company, and also for progressing the pipeline of either the drugs or products. So you have to be very strategic when you raise your Series A, number one. Number two, what investor looks when they invest into the company as a Series A funders? Basically, they would like to see some sort of product or services, whatever you are offering, at a little bit mature level. Preferably, some returns on investments from the seed funding is always helpful. And if your balance sheet is positive, then at least you are on the negotiating terms. So I would say that you could delay Series A as much as you can initially, and you should not start a company just to raise funds. Raising funds is basically to make the company successful. So that should be the bottom line. Does
Thank
that
you.
answer the question?
Yes. And sir, here is the last question, and that is regarding the student entrepreneurs.
Yes.
So, unlike the independent entrepreneurs who have a committed time to explore the entrepreneurship, and unlike the faculties or other researchers who have basic infrastructure to explore the entrepreneurial path, how does a student enters this path with the tight academic schedule, which don't leave them with the time availability, and they lack the basic resources which will be needed to start this very journey? So, what is your suggestion to the aspiring student entrepreneurs?
So in terms of basic resources, I think that is where the incubation center chips in, where they can provide the basic infrastructure. And when it comes to student entrepreneurship, you will always find that the faculty members would be happy to host you if you are trying to do something substantial in terms of product development. So it's again, pitching is a very key component of convincing people, right? So you'll have to pitch your idea to the incubation center in the same way you'll have to pitch your idea to the people who can provide you resources. And I don't see that as a big challenge, really. Now, being a student, there's no way you can invest all your time into studies and then also have additional time to invest in the entrepreneurship. So the time remains the same. You have to figure out how do you balance between the two, right? Which also means that depending on the individual and their priorities, they might or might not decide to go for such pursuit at the time of studies, right? So fourth year is, if you're a BSM student, then up to fourth year is when you are doing all these courses, right? And then the fifth year is something where you are already into your research project, which can, if you have an idea that you can couple along with your research project, then it kind of gives you a win-win situation, right? You are working on a problem that can translate at the end of that year, and then you can start looking for the grant in aids and other things. So that's one way to think about it, but staying mentally prepared and think from the perspective of an entrepreneur would help you in the long run. Stage can differ. So that's all I would say. Sandeep, there's one more question that I noticed. Are there indigenous competitors involved? This is from Ashwin.
Yeah, yes.
So Ashwin, for the antibody drug conjugates, no, there are no indigenous competitors. In fact, there's a company I would not name because of various reasons, who reached out to us where they were setting up their biologics division, and they, even after COVID-19, they just initiated it 2 months back. And that is when they also did the Indian science and market scenario evaluation, and then they realized that, okay, it's just I guess them and us who are looking into this system at that level. So that's what led to our partnership. We decided that we will go together. But in the global competition level, yes, Japan, US, Germany, even Canada, they are investing big time into this. Means in France and other countries as well. So Roche and all those things, all these big companies. Think about any big company, and they would definitely have a portfolio in that direction, but not necessarily the technology to take care of the precision. So that's where we have the edge over the global competition.
Okay, so this ends our Q&A session. And let me take this opportunity to thank Dr. Vishal for the wonderful talk and for sharing his time and experience with all of us. I would also thank IISER Bhopal for the support in organizing this event. I would like to thank Dr. Amjad Hussain, CEO of ICE, the ICE staff members, and especially the student innovation leaders who helped us in coordinating and conducting this event. And finally, I thank all the participants to share your time, and I hope you really enjoyed this talk. Stay tuned for the next event of IISER Bhopal. Until then, stay safe. Thank you.
Thank you, everyone. Stay safe.